Hello everyone. I hope you are all well and getting the hang of doing this class online. It certainly isn't the way I would prefer to do it. With this PowerPoint, we are starting the material in Module 4. So there'll be a couple PowerPoints, this and genetics. There'll be Canvas quizzes, a writing assignment, and an exam at the end. So this is the first of several PowerPoints on the material on Module 4. In this PowerPoint, we are going to begin talking about meiosis. And we previously talked about mitosis. And mitosis is the method used for making copies of somatic cells. And somatic cells are all body cells except for sperm and egg cells. So in this PowerPoint, we're going to talk about meiosis, which only applies to sperm and egg cells. So that only applies to reproductive cells. Mitosis is used for all other cells. You may remember we talked a little bit that all your somatic cells, that is all your cells except for sperm and egg cells, have 23 pairs of chromosomes. So they're called diploid because they have, every chromosome comes in pairs. So we have 23 pairs, which gives us a total of 46 chromosomes. So you got 23 of your chromosomes from your mother and 23 from your father. And that gives you the total of 46. And that's what makes them diploid. This 2N refers to 2 of every cell chromosome. That's what the 2N refers to. And the pair that you got from your parents are called homologous chromosomes. That's one from your mother, one from your father. And that's what makes up homologous chromosomes. In meiosis, we are using a process whereby we're starting with a diploid cell, which is what all of your cells are, except for sperm and egg cells. And if you think about it, if you had a sperm cell with 46 chromosomes and an egg cell with 46 chromosomes, and then when fertilization occurs, you would now have 92 chromosomes, which is too many for human cells. So the process of meiosis is used in making sperm and egg cells, and instead of having 46 chromosomes, they're only going to have one of every chromosome, so that they're going to have 23 chromosomes. And having only one of every chromosome is called haploid. So haploid is when you have just one of every chromosome. So that's what that term refers to. And another name, as we start genetics, there's a lot of terminology. But sperm and egg are also called sex cells. They are together referred to as gametes. And so we'll be using that term a lot. So meiosis is the process for making gametes. So if we have a sperm and egg cell that each have only 23 chromosomes, then when fertilization occurs, we have 23 in the egg, 23 in the sperm, and then we get back to our normal 46 chromosomes. So meiosis is a way of keeping the chromosome number intact after fertilization. So again, this is kind of just showing that. So say this is one chromosome from your father or mother, and one from your other parent. And the pair of those are called homologous chromosomes because they're identical, one from each parent. And we talked last time about how during the S phase of the cell cycle, you make a copy of each of them. And those are called the sister chromatids. So that's the one from your mother and its identical copy, and the one from your father and its identical copy. So the copies are called sister chromatids, and the pair are called homologous chromosomes. That's always a little bit confusing, keeping those straight. So before meiosis, we go through the cell cycle, and we do make a copy of all the chromosomes so that you do have the sister chromatids for all 46 chromosomes prior to beginning meiosis. Okay, so once again, meiosis only occurs In reproductive organs, which in humans are the testes and the ovary, excuse me, they only are used for making sperm and egg cells for sexual reproduction. Another special thing that happens in meiosis is that you get what's called gene recombination. So you have a chromosome that you got from your mother, one from your father, but those genes are kind of mixed up so that the egg and sperm has a combination of your parental chromosomes. So at the end of meiosis, you're going to get haploid cells, which means they only have one of every chromosome. So in order to do that, meiosis goes through 2 rounds of cell division. In mitosis, we only had 1 round, but meiosis, we have 2, and they're cleverly called meiosis 1 and meiosis 2. So you start off with your normal diploid cell. It goes through meiosis 1. You get 2 daughter cells, each of which are haploid. Then each of those goes through meiosis 2, and at the end you get 4 daughter cells that are all haploid. So you start off with 1 diploid cell in meiosis, you end up with 4 haploid cells that have 23 chromosomes in humans. So I'm just going to quickly go through the stages of both meiosis 1 and meiosis 2. And they have the same names as the stages of mitosis, but since there's 2 rounds of divisions, they're called, in meiosis 1, the stages are called prophase 1, metaphase 1, anaphase 1, telophase 1, and then cytokinesis. So in meiosis 1, there's a couple unique things that happen. One is during prophase 1. So a lot of it's the same as mitosis. The spindle forms, the nuclear envelope dissolves, the chromosomes become visible as the little X things. But there's one important thing that happens in prophase 1, and it's the only time it ever happens. It's called crossing over. And that's also called synapsis. That's what that's for. So I'm going to go to the next slide and show you what crossing over is, and then we'll come back and finish this up. So after prophase 1, the cell then enters into metaphase 1. And in metaphase 1, what lines up are homologous pairs. So you have lined up along the center of the cell are the homologous pairs, one from the mom and one from the dad. And they're lined up. And then in anaphase, the homologous pairs of chromosomes get pulled apart. So the one from your mom would go to one side, the one from your dad would go to the other. They still have their sister chromatids attached. And then in telophase, the spindle dissolves, the nuclear envelope reforms, and the cell splits into 2 daughter cells. And they have 2 of every chromosome, but it's because they still have their sister chromatids. They don't have a homologous pair anymore, but they have the 2 sister chromatids. Then these 2 daughter cells at the end of meiosis 1 will both go to meiosis 2. So here's one of the daughter cells, and then here's the other daughter cell entering into meiosis 2. And then in meiosis 2, they go through those same 4 rounds. So prophase 2, metaphase 2, anaphase 2, telophase 2. And so in meiosis 2, again, the spindle forms and the sister chromatids line up in the center in metaphase. One sister chromatid goes to one side, the other sister chromatid goes to the other side in anaphase. And then in telophase, the nucleus reforms and cytokinesis occurs and the cell splits. So at the end of meiosis 2, you have 4 haploid daughter cells. Remember in mitosis, you had 2 diploid daughter cells. And the other thing, because of the way the chromosomes line up and crossing over, that they're all genetically different. So one cell could get more of one parent's chromosome than the other cell. So you would have an egg that has more of the father's versus the mother's. So it's a way of getting genetic variation by the way those chromosomes line up during metaphase. And it's that crossing over and the lining up of the chromosomes that leads to variation in offspring, which is shown here. So you have a dad and a mom, pup, dog, and you can see all 4 puppies look different. And that is due to the fact that you have variation in sperm and egg cell productions due to recombination of chromosomes and crossing over in meiosis. And just to give you an idea of what kind of variation is possible, because of the random separating of your parental chromosomes, that gives you this many different possible combinations in a zygote. A zygote is another name for a fertilized egg. So that's a fertilized egg. Fertilized, which I just spelled wrong. And so that's a really big number. That's 70 trillion different possible combinations. Then when you add in crossing over, you get even more possible combinations. So that's how you can get so much variation in offspring. It's due to the process of meiosis causing new combinations to occur of genes in the offspring that did not occur in either parent. I'm not going to go through this slide. I kind of put this more here just as a summary for you of the differences between mitosis and meiosis. And again, meiosis, there's 2 rounds of division. There's crossing over. They're haploid. You get 4 daughter cells. Each of them are different. And it only occurs in for sexual reproduction. So again, this is the process for making sperm and egg cells. And so during fertilization, a sperm and eggs are made by meiosis. They join together at fertilization and form a zygote, which is the embryo. So it starts out as one cell, which is now diploid because you had a haploid egg and a haploid sperm forming a diploid embryo. And then you undergo mitosis from that point to form the adult organism. And that's just shown here that in a male and a female, the process of meiosis produces a haploid egg and a haploid sperm. Then these haploid sperm and the haploid egg join together at fertilization to form a diploid zygote, which goes on through mitosis to form the adult organism where all the cells are diploid except for sperm and egg cells. I just wanted to mention that in humans, the timing of meiosis is different in males and females. In males, which meiosis produces sperm, so the process of producing sperm is called spermatogenesis. This occurs in the testes, and once a male enters puberty and begins making the hormones to stimulate sperm production, it occurs continuously in the testes. We'll talk a little bit more about this in detail at the end of the semester, but these are the diploid cells, or called spermatocytes. They're in the testes, and they undergo meiosis to form each diploid cell that starts out forms 4 haploid sperm. In women, the process is quite different. So there is a oocyte, which is the egg cell. And you may have heard people say that you're born with all your eggs, and that's actually true. The way it happens in women is that the primary oocytes, which is the diploid cell, kind of the parent cell of your eggs, they undergo meiosis embryologically. So that is before a female is born. They begin meiosis, and it stops partway through meiosis 1. So when a woman is born, she has all of her egg cells that she's ever going to have are partway through meiosis 1. And they stay kind of arrested in meiosis 1 until a woman gets to puberty. And then under the influence again of reproductive hormones, every menstrual cycle, one of those eggs that are arrested in meiosis 1 gets stimulated to complete meiosis 1 and enter meiosis 2. And then they only complete meiosis 2 if it's fertilized by a sperm. If the egg is never fertilized, it actually never completes meiosis 2. And it's expelled in menstrual flow without having ever completed meiosis 2. The other big difference in female egg production is that they don't actually form 4 haploid daughter cells. So during meiosis 1, there's only one big cell formed, and then the other cell kind of is not given all the good stuff of cytoplasm and mitochondria and all that. So it kind of just disintegrates. It's called a polar body. Same thing happens in meiosis 2. So you have another polar body. So at the end of meiosis, you only get 1 haploid cell versus 4 haploid that you get with sperm production. And one of the reasons is that the egg is a much, much bigger cell, has lots more stuff in it. It's really the source of nutrients and growth for the developing embryo. So whereas sperm you produce by the millions and most of them are never used, eggs are produced a little more frugally because they are a bigger investment in mammals to produce an egg. So we don't have lots of eggs that are wasted. You're still born with many more eggs than you need throughout your reproductive years. So it's not like you don't have enough eggs and you can't make any more. You have thousands more than you'll ever actually use. I think this PowerPoint's running longer, so I think what I'm going to do is I'm going to stop there and kind of split it into 2 PowerPoints and continue on with some of the other